Hi, everybody. Thank you so much for being here. My first Smithsonian's Botanical Symposium was back in 2012 when I was a PhD student at George Mason University, and I never thought that I would become a speaker in the symposium, so I'm very honored to be here. And we've been hearing for 2 days now about symbiosis, and lichens are a classic example. However, the way we see lichens has substantially changed over time. Before, it was considered a dual organism of a fungal partner, or the microbiont, and a photosynthesizing partner, the photobiont. Now, this concept has extended to also include a diverse community of microorganisms. And you probably heard about yeasts being present and impacting lichen phenotypes, for example, but not all lichens have them. But as other organisms, bacteria have been shown to be commonplace in lichens as well, which along with possibly then other fungi, compose the lichen microbiome. And only when all of these components come together, we see in nature this structure that is resulting of the symbiosis, the lichen thallus. The microbiome is providing the thallus with a structure, protecting the algae from the outside environment, so providing a much more stable long-term environment, as well as water control and mineral nutrients. And in return, the photobiont is sharing with the microbiome its products from photosynthesis, and therefore sort of providing the food. And the microbiome, which is composed, again, of primarily bacteria and fungi, can have several functions, such as provision of hormones and vitamin B12, resistance against biotic and abiotic stress factors, among several other functions, most which are unknown. And due to this added multiplicity and unique symbiotic structures, lichens have been described as mini ecosystems, holobionts, or meta-organisms. So what is the lichen dilemma? The dilemma is then how to explore and properly assess diversity in lichens, given that there is just so much going on. And for that, then we need to know who is there and why. And only then we can evaluate who are truly symbiotic partners in such complex symbiotic systems and who is just hanging out. Today, I will present some results to my lichen dilemma, utilizing examples of this subtribe, Dictyonemateae, a group of basidiolichens that is lichen-forming fungi in Basidiomycota. These lichens are in the Agaricales order and more closely related to most mycorrhizae and saprobic fungi than to most lichens, for example. The photobiont of this group is cyanobacteria of the genus Rhizonema, known to be chiefly lichenized, but very properly for this symposium, they have also been found inside pockets of Frulania liverworts. And just by looking at these images, we can see an ample diversity, right? So just different colors, growth types, arrangements, and so forth. However, this lichen group was then for a long time thought to be composed of a single genus and 5 species, a concept introduced by Parmasto in an attempt to organize this a mass of over 40 names in 9 different genera. Parmasto monographed the group, synonymizing everything into a single genus and with 5 species. And as taxonomists, we love being out there in the field collecting and bringing these specimens back and putting names. And this system did simplify identification. However, with so many few names available, many different forms with distinct ecological preferences were not considered and dumped into this, what we call a taxonomic trash can. And all foliose forms under the name of Dictyonema pavonia would fall in there, you know. So if you collected, so this is a group primarily found in the neotropics. So if you collected in Puerto Rico or in Patagonia, it would all belong to the same taxon. And very similarly to filamentous forms, which then had 4 names, but primarily one neotropical, Dictyonemateae. So again, they would all be identified as this. But we were out there in the field noticing all these differences. Could it be possible that these differences are indicating possibly more than just a couple of species? And this is when I started researching my main question: how diverse is this group? In order to assess global diversity of the group, we did a lot of field work ourselves, but also study collections deposited in herbarium, as well as we counted with a lot of collaborators who would send us samples. And for the systematics part of this study, I utilized the concept of integrative taxonomy, gathering as much information as possible for each specimen. And for the microbiome work, I am also taking an integrative approach. So what I do is I sequence everything for amplicon, and then also for selected specimens, do metagenomics, fluorescent in situ hybridization, short is fish, and then also some cultures. But I will present here primarily and just the amplicon data, which is what we have corresponding data. So the same set of samples that we have microbiome, photobiont, and bacterial microbiome data. So this is what I'm going to present as in this part now, the results. And it comes as no surprise that there is an enormous amount of hidden diversity. And I always think that first we need to establish a base knowledge regarding the fungal species, which is what is naming the lichen. So we only when we have very well delimited species boundaries, we can, you know, have a better understanding of these symbioses, and then of course also of their evolutionary history, which was definitely not the case when I started. So we found that one genera was actually 5, based on phylogeny, ecology, and anatomy and morphology. And we can see that both filamentous and folios, each forms, which were only a few names as we saw before, are now 2 genera each, and Microsquamulous genus, Cantholichen, which in all fairness was discovered by a group decades after Parmasto's monograph. So it was not included back then. And this is the first work that we had out showing these 5 genera and their relationships. And Cyphilosterium, here in this beige color, is the earliest divergent group and sister to the rest of the clade. Then there is this paraphyletic grade, Dictyonemateae, representing a transitional morphological and anatomical composed of, you know, these forms. Then a derived clade here with 3 monophyletic genus, Acantholichen, Corella, and Cora. As you can see, this paper is a bit old now, so I'm going to move on to a more recent paper. And we have data, microbiome data for almost 2,000 samples. But as I mentioned before, I'm just going to present the part that we have corresponding data for the photobiont as well. And the colors that we now see in the background represent then these 5 genera. And these are more or less the numbers of species in each one of these genera. And it's just amazing that from just a couple or 5 species, now we have over 300. And interestingly, these 5 genera, they were all proposed before. So we are able now to confirm with molecular data and resurrect this genera. And same pretty much for all of the 40 species names that were available before. And we also found that this group actually has a very high level of endemic species, as opposed to species that have very large geographical distributions. In fact, only, I don't know, maybe 5% of the species, of these 300 species that we found, are actually widespread. And one thing that kept us wondering, though, was this split here in Cora. However, due to their very uniform morphology and anatomy, we accept as a single genus. But now that we have a much better understanding of the species in this group, we can move on to the photobiont diversity. So everything that we saw then regarding the microbiome was this high amount of species that we didn't know of. And now switching to the photobiont, which I mentioned before is cyanobacteria of the genus Rhizonema, we see a very different result. We performed a network analysis, which help us to better visualize this divergence, given that the genetic marker that we use, 16S, is very conserved. It's still the most widely used for bacteria, but it's also valid to mention that we did do RBCLX, and we found similar results. So I'm just going to show part of this. Again, going back to networks is also nice because it accounts for the frequency of each one of these haplotypes. And then we can see then these larger circles means that there are more haplotypes in our sampling. And we found 3 putative species here, 2 monophyletic ones. So in this dark yellow or mustard color, Rhizonema neotropicum, and then in green, Rhizonema andinum, and then in this Rhizonema interruptum, just sort of this blue color, there's a lot more haplotypes and then also more diversity. So this could actually be a species complex. And within this lineage here, we detected 4 main haplotypes that received special colors. And we'll see these colors in the next couple slides. So bear with me, but I also include some cheat sheets along the way. So then next, we wanted to see how these photobiont lineages are distributed across the different lichens in Dictyonemateae. So I created ITS trees for each one of these genera inferred by maximum likelihood, like this one, that you see topology of the microbiont. And in this case here, Cephalosterium, which was the earliest divergent group. Then I mapped the different 16S haplotypes into this tree, and this is what you see. And I have Cephalosterium here, as an example. And as you can see, for example, the lineages, only one is present here. And again, the pink and the purple are some of the most common haplotypes here. And if it just has this royal blue color, means it's still within this lineage, Rhizonema interruptum, but one of these less frequent haplotypes. Now, I want to include the entire clade for you to see how these photobionts are distributed. But so instead of looking genus by genus, we're going to look at this circular tree. And you remember this from before, right? Now, in addition to the genus colors, which are in the background, I will plot these haplotypes. And this is just then now the distribution of the different haplotypes in the fungal tree. However, there's just too many colors, so we can simplify then this tree to just show the 3 putative species, which is now, as I said, simplify here. So we can see that as a subtribe, Dictyonemateae has all lineages. However, now it's more clear that what was causing this split in Cora here, so Cora clade A and B that we saw before, is this a switch of photobiont choice in the common ancestor of Cora. So you see that this Rhizonema andinum here is primarily found in only this Cora clade A. And other conclusions then from this is that there's very low diversity of the photobionts with only 3 lineages detected. And this diversity, clash between, microbiont and photobiont can be explained by evolutionary constraint posed by lichenization, where many species and genera of lichens share the same optimal photobiont in a process very similar to how we deal with crops and agriculture, right? So the best ones, are being widely shared and most consumed by us, for example. In return, the photobiont then it's not able to diversify as much, but I can still see this as successful exactly for the same reason, due to this domestication, because now this partner, this photobiont is able and has managed to sort of get the best out of the situation and is now being shared amongst many different and unrelated groups of fungi, because rhizonema is not only found in basidiolichens that I'm showing you here, it's also found in several genera of ascolichens, so lichen-forming fungi in Ascomycota. And I think that's something worth to mention, again, because of the plant symbiosis topic here, is that these ascolichens have been the ones that there's research showing then that, so remember I mentioned that rhizonema is able to live inside of these Rholenia pockets. So close to these Rholenia pockets with rhizonema, guess who was living in close proximity? This some different genera of ascolichens with this same photobiont. So not only rhizonema has managed to be a partner with different lichens, and so completely unrelated in basidio and ascolichens, but also with bryophytes. So let's talk a little bit more about this. As you can see, I get super excited, right? So in the previous slides, we saw that the same lineages of cyanobacteria found among many species of lichens, but this is not only something that you see in the DNA, you can also witness this in the field. And in this image, we can see photobiont associations with, sorry, I got a little bit distracted here. So here we see in the field Dictyonema and Cora living together, but then going back to the DNA, this is then an example how we can compare which samples of microbiome are being selective or specific in this case towards the photobiont. And then in the highlighted here, you can see that the same lichen can have different haplotypes of photobionts, but it's always within the same putative species of rhizonema. And finally, then switching to bacterial communities of these same lichens, I wanted to show you this principal coordinate analysis results, which is a method to explore and to visualize similarities and dissimilarities of data based on this distance matrix. And then each sample is assigned a location in a 3D space. And each one of these samples then is a circle, and they've been color-coded according to the microbiome genus they belong to. And we can see that there's a suggestive pattern in bacterial communities present in the different genera in Dictyonema, with the earliest diverging group, Cephalosterium here, showing the most unique community, but still more or less similar to Dictyonema, which is transitional, to a more unique community in Cora. And taken all together, then there seems to be a phylogenetic correlation with the diversity and structure of these microbial communities, in addition to ecological preferences and growth types. So diversity of lichens can be studied in different levels. I always think as a basis, we should have very well delimited species of the fungi, so we're able to compare apples to apples. And in the Photobiont, we can assess how diverse they are and how selective and specific they are towards their choices, but also what microbes are present. So now that I know who is, present, my next part of the dilemma is design novel ways to test and evaluate what roles they play and how essential they are for the lichen symbiosis. For example, what these microorganisms are doing there and to which level they are required for the functionality, stability, and survival of lichens to be able to really say real partners in this multi-species symbiosis. With that, I would like to thank all my colleagues as well as multiple funding sources. Again, I would like to thank the organizers of the Smithsonian Botanical Symposiums of 2021 for the invitation and you all for being here. Thank you very much.